Thank you for this invitation to come and talk to you about our beta testing of the version 3 of OncoScan, and happy to be able to share our experience with this and maybe also a little bit of how the data looks and what we've learned. So it started by saying, okay, there's a number of FISH probes that we commonly run within ARUP. We used 4 20-micron sections for our DNA extraction. Actually got a lot more DNA than we mostly needed, but that was what we started with. We estimated the tumor percentage on a section that was cut after the sections for the DNA extraction. We did not do any microdissection because our percentage of tumor ranged from 40 to 90%, which was high enough to not require microdissection. So we'll start with the gliomas, and I just wanted to start with this little flow chart that kind of represents what are some important molecular diagnostic markers in gliomas. And so what you'll find is that one of the first separation points would be a mutation in IDH1 or IDH2, versus over here an EGFR1 amplification. Then you can further subdivide, these are according to the WHO categories, but you can further subdivide 1p19q co-deletions, and then looking at CDKN2 on 9p loss and 10q loss, which is selecting for a p10 alteration. And so if you have a single test that can help determine the IDH1-2 mutation status, the p53 17p status, the 1p19q status, the 9p, the EGFR, and the 10q status, you can really do a nice job of just jumping down this chart to identify what grade your glioma is at. So the EGFR1 positive cases, I've just put this flowchart at the bottom sort of to remind you what we're looking for. We already knew by FISH that all should show amplification, and by OncoScan it was not subtle at all. I'll show you that data. All showed a 9p deletion involving CDKN2. Some were very, very focal deletions. All showed a loss across 10q, so it would involve p10. None had IDH1 or 2 mutations, as you would expect, because they're all jumping down this pathway. And none had the 1p19q loss. So again, we had many lines of evidence that really suggested or confirmed that this was a primary glioblastoma grade 4. Here's what the data looks like in Nexus. This might change a little bit from the final version of the software, but this is your B allele frequency up here, and this is your copy number log-2 ratio. Here's EGFR, so if you could not see that was amplified, then you might need to get glasses. But it's really obvious to see that amplification. And then you can also see, of course, many other chromosome copy number changes, and you can get confirmation of those copy number changes with the B allele frequency, and I'll also show some slides in a minute that shows that copy neutral loss of heterozygosity that can be informative. And then here's our very focal biallelic CDKN2 deletion, and here's our loss of chromosome 10, including P10. Here's another sample. I'm just going to point out again that we can really easily see that EGFR amplification. And then here's an example of copy neutral loss of heterozygosity here on 7q. So you have the amplification here on 7p, and then here on 7q it goes back to a normal copy number. But here you can see that allelic imbalance demonstrating loss of heterozygosity, copy neutral. For the 1p19q deletion positive cases, they were all positive by FISH, they were all positive by OncoScan. And one of the nice things about getting the sizing is that we expect these to be whole arm losses because of the mechanism of it being a whole arm translocation with then loss of one of the derivatives. And so you really could be sure that you were really looking at the type of 1p19q that you expect. And that size matters because you can see partial 1p deletions in glioblastomas without the 19q, but they're not whole arm, they don't have the 19q, and they have the opposite prognostic value. So getting that sizing can matter. All 9 were positive for IDH1 mutations. IDH1 is more common than IDH2 in this subset, and all of ours were positive for an IDH1 mutation. None were positive for EGFR1 amplifications, none were positive for 10q loss, and none were positive for 9p loss. So again, now we had multiple lines of evidence to help us know that we were here at grade 2, and that we should have a good prognosis and expected response to therapy. Here's what some of the mutation data looks like when you kind of narrow in on it. So what it can do is take all of the samples within that run. So this run had many different tumor types in it. 4 of them were 1p19q deleted. There's 2 right here that are sitting on top of each other, so they look like one star. But so these are all of the samples in the run. This is controls that are put in the run, 3 negative controls and 1 positive control, positive expected 20%. And then this is an in silico reference set. And so for this particular mutation, you can see that these 4 1p19q deletion cases have a signal intensity that is consistent with the positive control for this particular mutation. And all of the other samples, which were not 1p19q, tracked with the normal. So high confidence that that mutation exists. All right, here's the 1p19q data plot. So this case is actually pretty calm. So here's your 1p loss, here's your 19q loss, confirmed by also the B allele frequency, and the rest of the genome is actually pretty copy number normal. Here's another case where again you see the 1p loss, the 19q loss, and then you can see a subclone, that 18q loss, and there it is with your B allele frequency. You can see that by the deviation pattern that you've probably got this in a subset of cells, somewhere around maybe half or maybe a little bit less than half of the cells now have a subclone with an 18q deletion. All right, so the lipomas that were MDM2 amplified, all 10 cases were positive by FISH, all 10 cases positive by microarray. For the cytogeneticists, I'm sure you all are, you remember that a lot of times this happens with the ring chromosome, some type of large marker chromosome, and so we think that there are other regions that are co-amplified within that marker, and that's also what we saw with OncoScan. We would see an amplification around MDM2, but we'd also see amplifications of other regions of the genome. Many of them were also on 12q, but sometimes they were in other chromosome regions, so some other material that may be jumping into that marker chromosome and co-amplifying. This I also chose because it's another thing that you can do with this platform as you're looking across the whole genome is to try to figure out what is my exact ploidy and do I need to do some type of recentering to really get an accurate copy number, because we know that sometimes increased ploidy can then during the normalization process throw off the actual copy number. But what you can do is look across here and for a pattern that you expect from your B allele frequency and a pattern that you expect from your copy number and do a matching game to identify where's my true diploidy. And so here's an example of where the copy number on chromosome 10, 11, and 13 looks like it's probably all about the same, but the allele pattern says, no, clearly there's something very different. 11 and 13 do look like each other, but 10 actually has a much more normal pattern. And so is 10 really normal, and then these are copy neutral loss of heterozygosity? Well, I also, don't just look at a couple chromosomes, look across. 2 also has kind of a normal B allele frequency pattern, but looks elevated, whereas 10 looks lost. So what's really normal? Do I need to kind of bring the whole genome down or bring the whole genome up? And sometimes with only B allele frequency, it can be hard to decide if you need to go up or down. And so one of the things that this platform is going to have when you look at it in Nexus is both your allele peaks and your B allele frequency. And so some of you are like, I have to think about 2 ways of looking at these patterns, and believe me, it's not so bad. You just have to remember one is A minus B and the other one is the percentage of B. But when you get used to that, then what you can recognize is that chromosome 2 is spanning out on your allele peak, so it is really gained, whereas chromosome 10 is having a normal pattern by allele peaks, and they're both having normal patterns by B allele frequency. So chromosome 10 is our normal diploid chromosome, and now we can recenter on that. So I think it's powerful to have both of these allele patterns to then really do appropriate ploidy detection. All right, so our neuroblastoma that was MYCN positive, it was also positive by OncoScan, not surprising. Here it is over here. Again, you'd have to be, not looking to not see that. Plus, you can easily see some of the other things that we know are useful for subdividing neuroblastoma. So this is just taken from the Atlas of Genetics and Oncology, and it's just showing that, by determining both ploidy, 1p, 11q, and MYC amplification, you can do a nice job of differentiating favorable versus aggressively behaving neuroblastomas. And for the breast tumors, so we had 6 cases that were HER2 amplified And only 4 of them confirmed, and that's actually ended up being some of the most interesting data to us. So here's one of the cases that did confirm. So you can see that there's copy number alterations throughout the genome, but here's where HER2 is located on 17q, and you can see the little spike right there. So, but what about, you're all interested in the 2 that didn't. But before I get to that, let me just also mention that having that very focal copy number information can be useful as well, because one of the questions that is now becoming more common to ask is, what else is co-amplified? Are there other things that are targetable? There's a nearby gene to HER2, TOP2A, which if also amplified, it may indicate that that could respond to topoisomerase inhibitors, and it's very close by, so it often can get co-amplified. But here's a case where you can see, here's HER2, and there's its real Hugo name, ERB2. Anyway, so here is HER2, and you can see that it's amplified, and TOP2A is right next door to RARA, so here's TOP2A, and you can see that it's not amplified. So in one assay, you can give that information, which we're getting more and more requests to provide that information. And here's one where you could say that it is co-amplified. So here's HER2, and it's in an amplified region, and here's TOP2A, and it's in an even more amplified region. Okay, so what about those discordant cases? Well, here's the first example. So it's not that we just didn't have a tumor burden. We really do see easily that there are copy number alterations across this genome, and with a consistent B allele frequency pattern. And here's chromosome 17, and we'll do a zoom in on it. So a lot of you are going to immediately recognize that 17p is lost, 17q is normal. But the way that the FISH is done, it's a ratio. It's a ratio comparing the HER2 to the centromere of 17, but the centromere probe is not smack on the centromere. It's usually satellite sequence adjacent to, and it's more satellite sequence adjacent to the P arm. So if you have a relative loss of your control, then your HER2 is going to look falsely amplified. And that's what happened in this case. But by having the whole genome data, you can easily recognize that this is actually just a 17p loss, not a 17q gain. Here is the other discordant sample. Again, we obviously do have a number of copy number alterations that are occurring. In this particular case, the FISH was signed out as, it was not informative, they couldn't be sure. They reflexed to a PCR assay that we have that they did call just above our cutoff for amplified. We put this on the OncoScan, and again, here's 17, and what is 17 doing? It's actually a loss of 17. It's a monosomy 17 across. And here are the HER2 probes, and they are a little bit less lost than the rest of the chromosome, but it would be wrong to call that amplified. So we asked well, is this known? Well, a quick PubMed search said, yeah, there's already data out there that shows that false positive FISH results can be due to relative loss around the centromere rather than actual gain of HER2, and it's estimated about 20% of the cases are false positives, and that arrays can be very useful in really giving you confidence in the correct answer. And so this was data from a paper that was published in 2010. They used a back-based microarray, so they unfortunately had to have a lot more DNA than we did in order to do this assay, but they had a number of cases that also showed that loss of 17p with a relatively normal or slightly gained 17q, but that loss of 17p was throwing off the ratio, and then also a monosomy 17 point throwing the ratio. So this is from their paper, and I think it can make good sense that if you have disconcordance between your immunohistochemistry and your FISH results, then it's really one of the best ways to resolve this is by using an array and getting that whole genome information. And then lastly, this wasn't part of the beta, but I wanted to bring it up because one of the questions that that comes up is, okay, we've looked at V1 and V2 from OncoScan. How does it compare to V3? And so we had also done studies on V1 and V2, and so we were able to come back to those samples and run them on V3. But let me at least just tell you quickly about one of the studies that we did on V1, and it was looking at using this platform to distinguish melanocytic lesions of uncertain potential from true melanoma. And of course, you can imagine that this is an area of great interest, that there's about a 25% discordant rate among pathologists for some of these challenging lesions, and so it's nice to have another assay that really helps you get more information about that definitive diagnosis, because I think when it comes down to it, if they decide they're in doubt, then they cut it out. And that may not sound so bad, except for if it happened to be like in a really prominent place on the body, because they do usually about a 2 centimeter margin. And so some type of testing that can help differentiate these lesions could benefit patients. It's not going to eliminate the need for all invasive procedures by any means, but it may reduce the number of benign nevi that are subsequently removed and then were benign, and give clinicians added confidence in the decision to proceed with treatment. And I kind of like that second one better, which is, if you're going to give me a disfiguring scar, I want to have some evidence that you really needed to do that. I'll live with the scar just fine after that. So there was already literature that showed that 95% of melanomas did have characteristic chromosomal gains and losses that you could pick up with microarray technology. And so this is just melanomas and gains in green, losses in red, and versus benign nevi. And Spitz nevi will occasionally show gains of 11p and 7q, but outside of 7q, that was the only minor overlap. And the melanoma usually had 5 or more chromosomal changes, whereas the Spitz nevi would have a single alteration. So that was felt to be a pretty good distinguishing factor. And so we went with 64 melanocytic lesions, 23 were benign, 11 were of unknown potential, and we had 27 primary melanomas and 3 metastatic. 98.5% passed the MIP V1 assay. We used between 6, these were our DNA yields, and actually one of these samples, you can't tell it necessarily by looking right there, but one of these samples had only 15 nanograms of DNA, and we went ahead with it. Thought, well, let's just see how it went. And it also really worked very well and could show clear gains and losses. And then our blocks ranged in quite an age, and this was recently published if you want the details. So in essence, we had very similar gains and loss patterns. So this is the historical data from Bastian et al., and this was our aggregate from these studies that from the 27 melanomas. We saw the gains and losses that were expected at similar frequencies to what you would expect. And you know, the literature had said that you would see in about 95% of cases. We saw it in 89%, but we did have a small sample set. And all of our benign nevi looked benign. I won't go too much into this, but this is just one of the problems, of course, with these studies is follow-up. Like, how much data do you really have to say whether or not those uncertain melanocytic lesions really did behave benignly? And because, again, it comes to this, because they were uncertain, they were cut out with 2 centimeter margins. So the fact that many of them have no evidence of disease may be because they were truly benign, or may be because they cut them out and they did the patient well. We did, I will note, have one that had a benign looking pattern, but then did have a positive lymph node subsequently. So there does appear to be one sample that we had that did have a benign pattern by the microarray and yet did act more aggressively. You may be aware of a FISH panel that is on the market. It is produced by Abbott Molecular and run through Neogenomics, this MelanoSITE assay. The samples are all run at Neogenomics, but pathologists can do their own interpretation through this collaboration that they may have. But that's the policy. The samples get run at Neogenomics. And so there's a part of me that thinks, well, I want to be able to run things at my own laboratory. And so I also thought, well, how would our assay compare to this assay that's now available, and there's lots of data on it. And basically this assay, again, since it's, oh, excuse me, I didn't mean to press that button. Since it's FISH-based, there's only a few loci that you can really look at. And you also need to have some control loci in there so that you can do ratios. So there's really only a couple regions of the genome that they're looking at. They're looking at 6p, 6q, and 11q. And then they're doing ratios between those 3 regions to identify positive cases. And in our series, looking at our data and asking, would we have had abnormal 6p, abnormal 6q, abnormal 11q? 6 of our 27 should have had a normal FISH assay because they did not have alterations of those regions. And yet they had alterations elsewhere in the genome that were consistent. Because remember, 10q, 9p, 7q, those are also recurrent. So here's an example of one of these cases. So you can see clearly that there's gains and losses that are ones that we associate with melanoma. There is a 6q gain, but unfortunately, here's the 6q gain, and here's where that FISH probe lies. That FISH probe is distal to the region that's actually lost in this patient. All right, so we felt really good about what this could do for melanoma and on V1, and how does it compared to V3. So it was the same blocks, just re-extracted. This is V1 data, this is V3 data. The patterns are reproduced, and the probes are acting a little bit better. See how much more, they're more spread out on V1, and they're tighter on V3. Sorry, I think I was saying V2, but yeah, so here's V1 compared to V3. We're getting a reproduction of the patterns and a tighter probe performance. So that's just one case, and then another case we've run now a number of them to just show that this was behaving very well. So, just wanted to acknowledge people within the laboratory, my scientists that are now really proficient at running this assay, collaborators within the Huntsman Cancer Institute and within ARUP that have been useful in helping kind of guide the thought process and really collaborating with to talk about the data. The ARUP Institute, which is funds us essentially, Affymetrix and Nexus for their development of this product. Thank you.